Hello student, last lecture we will study about bioremediations. Okay, and in this lecture we will study about microbial degradation of xenobiotics. Xenobiotics means xeno means strange, foreign, and biotics means living organisms. So let's see introductions of xenobiotics. It is the A xenobiotics is a chemical substance found within an organism that is not naturally produced or expected to be present within the organisms. It can also cover substances that are present in much higher concentrations than are usual. In other words, xenobiotics are chemical substances that are foreign to the biological systems or a compound found in human body which is not produced by itself. They include man-made or naturally occurring compounds, drugs and environmental agents. In degradations of xenobiotics, we learn natural compounds can also become xenobiotics if they are taken up by other organisms, such as the uptake of natural hormones by fish found downstream of sewage treatment plant outfalls, or the chemical defense produced by some organisms as a protection against predators. Okay, let's see types of xenobiotics. Xenobiotics can be categorized as a exogenous and endogenous according to their sources. But actually the main or basic types of xenobiotics is first one is natural and second one is a synthetic. So in natural we will learn about fungal classifications or bacterial classifications, that means bacterial toxins, mycotoxins, herbal toxins, human secretions. Okay, and in synthetic we learn degradations of drugs, pesticides, fungicides, chemicals, for example alcohol or methanols. And the classifications of xenobiotics is endogenous xenobiotics and exogenous xenobiotics. What is the exogenous xenobiotics? It is not foreign substance. They are synthesized in the body due to metabolic produce, that is a overproductions or produce in body. For example, bile acids, it is uh, produced in liver or stomach. Second is bilirubins in jaundice causes, then steroid, right? Next one is a exogenous xenobiotics. So in exogenous xenobiotics it includes various chemicals compound of origin such as drugs, environmental pollutants, food additives, hydrocarbons, pesticides, synthetic polymer and oil mixtures. It is totally foreign molecules ingested through inhalations or oral route and skin in the form of foodstuff, drugs, allergens and cosmetic. For example, carcinogens. Okay, it is a cancer substance, then drugs, food additives, then pesticides or pollutants. Next one is how xenobiotics are introduced in environment. Okay, first of all, xenobiotics, it is the formation of mineralizations. Then it produces carbon dioxide, hydrogen peroxides, water, inorganic salts, non-metal oxides. Then it also with the help of biodegradable process it is produces carbon dioxide, inorganic salts or non-metal oxides. Xenobiotics are processed out and to form the biotransformations, that means co-metabolisms. Okay, in biotransformations, the transformations of a non-growth substance in obligate presence of a growth substance or another transformable substance, and they release the substance changes from one chemical to another. And next one process is refilling, that means remobilizations. It is the modified organic compounds, less complex compounds, and it provide or to form the byproducts of retention in soil, sediment in water, release to atmosphere for the uses of biomass utilizations. They are also introduced to water bodies through industrial waste released. Okay, when these xenobiotics are released in environment, it need several years to degrade by themselves. So, in order to enhance their degradations, various microorganisms are used. It is the table for time required to degrade xenobiotics. This one biocides, and this sides is a time taken for the 75 to 100% disappearance. Okay, so first is a chlorinated insecticides. Their examples: first one is DDT. We know it is the time taken for 4 years. Second is aldrin for 3 years. Third one is the chlorodane for 5 years. Then heptachlor, it is degradations taken time is a 2 years. Then landant means a hexachlorocyclohexane for the 3 years. Likewise, next part is a organophosphate insecticides, and their example is a diazinon for 12 years, malathion for 1 week, parathion for 1 week. As like herbicides, for example 2,4-D, that means 2,4-dichlorophenoxyacetic acid for 4 weeks, atrazines for 40 weeks. Next one is a role of microorganisms in xenobiotics degradations. The members of the genus Pseudomonas, it is a predominant organisms for the degradations of xenobiotics or organic compound. So it is a soil microorganism are the most predominant microorganism that degrade xenobiotic. Different strains of Pseudomonas that are capable of the detoxifying more than 100 organic compounds have been identified. The example of organic compounds are several hydrocarbons, means phenols, organophosphates, polychlorinated biphenyls, means PCBs, and polycyclic aromatic and naphthalene. About 40 to 50 microbial strains of microorganisms capable of degrading xenobiotics have been isolated. Besides Pseudomonas, other good examples are Mycobacterium, Alkaligenes, and Nocardia. A selected list of microorganisms and the xenobiotics degrade is given in table. This is the table. This is microorganisms and polluter chemicals. For example, Pseudomonas species, Mycobacterium species, Alkaligenes, Nocardia, Arthrobacters, Corynebacterium, Bacillus, Candida, Aspergillus, Xanthomonas, Streptomyces, Fusarium, then Cunigammalia. For these, all organisms are degraded pollutant chemicals. Now we will study in next lecture the biodegradable pathways. Bye.